My conflict of interest slides. Just to give you a background, I have been at the Helmholtz Center, but since, 10 years now. I'm in Doha, in Qatar. And what we are interested here, I think if you don't know where that is, very close to Iran. I think John Chambers mentioned it already. I found it interesting this morning to hear there's a high percentage of diabetes in South Asians, and that he thinks it's not genetic. And I think we basically see the same in the Qatari Arabs, where we have 23% of diabetes. And our recent GWAS actually show that they are pretty close, actually, genetically to Europeans. So I think what we study here is a lot of things about the environment of diabetes. And of course, metabolomics is a very good tool for that.
Karsten, we don't have slides.
Let me go on with the slides for the introductions. I can do this very quickly because, we've seen this several times. We're interested, of course, in what makes us different. We know this is environment, genetics, the microbiome. It influences all kinds of complex disorders. And that's what we are very much interested also here in Qatar, especially, of course, diabetes and the comorbidities. I think we are going into the multi-omics era now really seriously. We will not only measure metabolomics, but also proteomics, transcriptomics, genomics, of course. But I will also mention later a little bit things like glycosylation, microRNAs, and epigenetics. I think especially epigenetics, as John Chambers pointed out, I must really share his love for these. And I will in my talk now present two halves. The first half, I wasn't really sure how to go with this because there was already Claudia presenting and Annette, and I think they all covered already quite a lot of things. So I went back a bit to the old slides we started when we worked with Suhre. But let me maybe first just finish this. I think the metabolomics as a middle endpoint within all these studies, I think we have seen that as well. So no need to insist here. What is really important, I think, is the cohort aspect. And we have seen that in Cora, we have seen that in UK Biobank. And of course, we have also a big cohort here in Qatar, the Qatar Biobank. And what these population-based studies actually can do is they identify risk for disease, complex disorders, links between the genome, the phenome, the gut microbiome, but they rarely reveal the call of pus phase and the novel biological understanding. And that's really where the omics technologies comes in. And the concept of the deep molecular phenotype as an intermediate phenotype between the cause of disease, the genetics, the lifestyle, the microbiome, and so on, and the disease endpoints, that's really the interactions between these different omics points. And that's what I want to talk about today. So let me just get into it. We started this off, and I think, so this is the slide I wanted to show. For those of you who don't know Jurek by face, so this is Jurek Adamski, and I think he will speak after me, and this is really why we do this similar today. And this here is a slide, I think even Jurek made it from 2008, taken at the metabolomics syllabus and in Munich when we really started off doing metabolomics. And Biocrates was really a big part at that time. So they were the first metabolomics platform that we actually used for doing GWAS. And I think this really was the first GWAS we ever did. And I was really a bit torn between my old talk to just present everything and all the big highlights, or just go into one case. And I think I will do this here. We actually discovered the FATS1 locus, the one that Claudia already mentioned, in our first GWAS in a very, very small study where we only studied 284 samples with 363 metabolites using the Biocrates platform. And let me just quickly go through this. So this was the first Manhattan plot. Actually, you couldn't even call it a Manhattan plot because the p-values were barely genome-wide significant. But as you see, there were already a few highly significant hits here, including FATS1. And so among the 5 strongest associations, there were actually 2 enzymes, FATS1 and LipC. I will only focus on FATS1 here, but LipC is a similar exciting story. So what we found for FATS1 is that this one associated actually very strongly with this phosphatidylcholines PC36 people working with Biocrates know this nomenclature. And we found something that at that time, when I worked with Christian Giger, he was telling me, normally you don't even show box plots for GWAS because you don't see a signal there. But the effect size was already very high here, like 10% explained variance by just one variant. So what is a PCAA36-4? And I think that at the time I really didn't know, and I think it's maybe good for those of you who are not so much familiar with the platform, is to go through this very quickly. And it also answers a bit the question, I think, FATS1, some people call it a pleiotropic locus, but in the end it's not. It's very precise action on one metabolite. FATS1 creates the fourth double bond in this polyunsaturated fatty acid pathway that goes from C18-2 over 18-3, then elongation step, and then comes this desaturation step. And this is really the enzymatic function of FATS1. And that's what we see in our data. Because if you ask what are the PCs, the phosphocholine, actually this is the pathway, the Kennedy pathway that creates them, where you actually have a glycerol molecule. There are fatty acid side chains added to that. It's a bit modified on the head end. And in the end, there is this kind of complex phospholipid coming with two side chains. And one of this you see here is, in this case, could be 23, could be anything else as well. So the PCs are important constituents of the lipid bilayer. And the thing is, you see all these different PCs, you find them in the Biocrates kit, like the 36.4, the 36.3, and so on. And what I highlighted here is, basically, we find all this associated with FATS1, but it's actually the enzymatic step between the 24 and the 23 that really is driven by this association. Here's just a quick picture of how they look like. And I think this is something I'm really a bit emotionally attached to. This is, you don't see much on this initially. It's just a plot of all the metabolites from the platform with a p-value and the estimate. And initially I had this in black and white on a Unix terminal. And I didn't really know, is this really something true or is there anything? Because we didn't have any metabolomics GWAS by this. But then what really struck me is when I looked at the effect sizes, like the directionality. See here, there's positive effect sizes, but there's only 2 or 3 double bonds. But if I go for the negative effect sizes, they are all negative. And then actually arachidonic acid, the product 23,4 from FATS is actually in the list as well. So then we asked, can we learn more about this? And I think this is a second really important lesson to take away from our studies with metabolomics, is something we also did at the Helmholtz Center, is actually looking at ratios. And at the time we had honestly too much compute power on the supercluster in Munich. I think it was the 10th fastest supercomputer at the time, and they wanted to have biologists working on it, not only physicists. I mean, they didn't know I was a physicist, but anyway, so we really spent hundreds of thousands of hours on that to do all the ratios against everything. And if you look at that, if you think about a biological reaction, a biochemical reaction, A gives B gives C with some simplified throughput K1 and K2. If you do some equilibrium study here, say, okay, the change in B is just what is produced and what is lost, and you make a steady state, which is valid in most situations, then the ratio between the product and the educt is just proportional to the rate constant, which would be actually proportional to the genetic variant in FATS. So I can translate that now into the equations. So here's FATS1, 24, 23 educt product pair. So actually the ratio between both is proportional to FATS1. And if there is a variation in FATS1 due to a SNP, it would be directly proportional to the ratio. And this is really what we found. So if we now look at ratios, so here's a ratio between the product and the educt pair. You find p-values before, remember, was 10 to the minus 9. Here we go to the 10 to the minus 22, which we would call today a p-gain, like the p-value would be 13 orders of magnitude stronger, which is extremely highly significant, even after correcting for all the multiple tests. And the explained variance goes to 30%. So you see also the box plot is much stronger. You really see it very strongly here, which you never saw at GWAS at the time. And also very interesting with the Biocrates platform at the time, they also had ether PCs, ethanolamines, inositols, and these, we didn't have anything significant in our GWAS initially, but when we went for the ratios, we actually found significant associations even for the PIs and the PEs. So I think that you could really learn a lot about this, and to coming back about this, to this idea about the pleiotropy. So you see it's always the ratio between 34, the 4 and the 3 double bonds. So actually everything that associates with FATS, there are lots of metabolites, but in the end, they are all carrying the same 23 and 24 side chains. So now I come to this overlap. I mean, in the future, we have seen that there were a lot of studies where you overlap GWAS and stuff like that. At that time, there was only very few, but I think you can already see the concept here. So this were the first GWAS with lipid traits, LDL, HDL, and we just asked, do they overlap? And the interesting thing is here, see, they overlap, but they were never reported in these papers. So these early papers didn't have FATS1 as a lipid locus, and we could actually predict that these would probably be lipid risk loci. And this was also done in another study. And then the same, I think this, I believe, is the first really big GWAS that came out in, I think, 2007. And see here, the coronary artery disease association FADS1 is now very, very strong, but at the time it would never have passed any significance level for a GWAS. So I think that was really very inspiring to say with intermediate phenotypes, you can really learn something about what's going on. And just as curiosity to see what happens today. So Claudia already mentioned Gabi Kastenmüller's group, and this is another server. So really write down this address if you can, sniper.org. So you can put in any SNP you have, and it would give you everything that is known around that SNP in an LD of 0.8. And here, the example actually is FADS1. It gives you information on that block, on EQTLs, PQTLs, and so on. And as I said before, so now here's the Lotta paper that Claudia just mentioned. So this data is in here as well. So you see here the p-value for FODS now is 10 to the minus 300. You can also overlap with that with UK Biobank, and you see also the HDL and triglyceride association we see before are much stronger nowadays, but they have been predicted actually in this study of 300 people. And really something curiosity, what I just found yesterday when I did the screenshots is actually here. You see facial aging comes up in UK Biobank. So I think we should really at some point do some kind of MR study to see whether actually omega-3 and omega-6 fatty acids, which are actually the product of FADS1, are causal for facial aging. I think companies selling creams would be very interested about that. Nap during the day, I don't know, but you can create your own hypothesis there and carry on from there. So I think that's really the interesting part about these studies. And going back to history as well, I mean, this idea of the intermediate phenotype. So this is a graph that actually Florian Cronenberg initially drew for our paper. And the idea of the intermediate phenotype, like linking the genotype through intermediate phenotypes to the endpoint, it's really very nicely shown in this degradation of the p-value. Very strong p-value for the ratio, which is really closest to the phenotype, to the reaction rate itself. 10 to the minus 8 for the lipids with a fatty acid in their side chains. 10 to the minus 4 for the HDL. The LDL as more intermediate phenotypes for the clinical one, and then the clinical endpoint like cardiovascular disease. And LipC, I think that we had the same thing as well. And I think in the meantime, we have seen tons of studies where you can overlap all this. Relates nowadays to keywords like Mendelian randomization, polygenic risk scores, and so on. So I think this is something I can only very quickly jump above, but I think what we really established in this first study already is what we call the genetically influenced metabotype. For FADS, it's very clear. So there's this variant, it's a variant in everyone. It's a very common variant. So it's not a monogenic disease. And in the end, I like to see this as a valve setting, if you want. Like every one of us is individual into these different valve settings, and the combination of all these metabolites really defines who we are. And I think this, let me just go quickly over this, goes to the same slide that Claudia already discussed, so I don't have to go back here and explain it again. So historically, once we did this, now let me go to the second part of my talk. Of course, we try to do more with this. So I want to very quickly just also give you a bit of a history. So this is the lab from Jurek Adamski, Cornelia Prehn here in the foreground. And in the back, you see this mass spectrometer with which it really started. So this was the first one we brought up together and where the first Biocrates kits really ran on. And after that, we had a lot of other studies going. Like we really used the kits in a bigger study, the work that we've seen before. Many of the data sets from core actually come from that study. We tested other platforms. We worked with urine, using NMR, of course, non-target metabolomics data. Numeris is something very similar to what Nightingale does nowadays. And also very quickly, so I thought, yes, I mean, this is a study actually Claudia is leading together with Adam Butterworth, and this is, to my knowledge, the most complex study we have nowadays. It's still not published, but I think the key message here is once you go to like the numbers here, 20,000 individuals, what you see is here again in this matrix, it's an example of different metabolites and different variants at the same locus. So now you can make these mini matrices and actually dissect the genetics at this locus and see which metabolites associate with which genetic variant. And you see here, for instance, 2 rare variants, 2 frequent ones, and the directionality is sometimes very different. So what you can really derive here is like, there could be variants changing the substrate specificity, pulling up or pulling down different metabolites, and others could go all the same way if there would be, for instance, variants in the transcription factor. All right, I think all this I can skip. Claudia already mentioned that. So in the interest of time, I would rather go into what I have to show new. So new keywords after this old slide was, of course, Mendelian randomization, polygenic risk scores. I think there's a lot going on here, but I won't insist in my talk here. And now as a teaser, I would like to show you a bit more about other omics coming in, being integrated also with metabolomics and other omics technologies. But before I do this, so now really take this more like a teaser to show you what you might be looking for. For instance, I have a database with all the GWAS done so far. So you can go here to metabolomics with an x.com, and there are basically all the studies I know of. If you know any, please let me know if anything is missing. And with that, I would like to move on. So you can already imagine where this is going, right? You can do genome against all of this. You can do this all against each other. So one thing we then did when I was here in Qatar, so very quickly, with my colleague Johannes Graumann, who was in Qatar at the time running the proteomics core. So we basically asked us, would we see the same thing in proteomics if we do a GWAS? And the answer is a clear yes. So what you see here is a GWAS with 1,100 proteins measured on the SomaLogic platform. And we find about 539 different protein QTLs that actually replicate. The interesting thing with the proteomics is it adds another level of complexity because there is the concept of trans and cis. So you may have variants that change one protein that interacts then with another one. So the GWAS hit could be in one protein's gene, and the signal could be on the protein encoded by another one. And for this, we also, I mean, this is a more novel one. So all these techniques of overlapping now, methylation QTLs and rare variants, and you name it, up to what really interests us is, of course, the association with the disease endpoints. And in this study, we found 83 different associations for 42 disease endpoints in the GWAS catalog. So quite a number of things to do. Also important to note, so we recently did a review of the FDA-approved proteins. There are actually 200 proteins that are now FDA-approved as analytes. And interestingly, 30% of them actually have a PQTL, which means that for 30% of these markers, you should actually take into account whether there's genetic variation in the background in clinical studies and so forth. And here again, just to be complete, I have a similar table. And the interesting thing is the numbers are almost where metabolomics stands, although that field started off much later. I mean, there's some early ones in 2008 as well, but I think proteomics is really catching up here. All right, then I said I'm trying to be a bit historic here because there is this link to what we did with Suhre. We actually did a study and I really would like to mention that here, a study we did in 2014. And this is a study I'm really a bit, I don't know, I mean, it really got unnoticed at the time. And nowadays with this epigenetic EWAS studies, it really becomes much more of an interest. And I just want to highlight something here. I actually remember doing this with Christian, and he was here in Qatar at the time, and we were really a bit like, oh, what's the story here? And at that time, we really didn't understand what's there. So we did an EWAS with the Illumina and the different metabolomics data sets we have in Cora. So this is a Cora study. So we have the Biocrates kit, the Metabolon, and the NuMar. So that's basically the Nightingale-like lipoproteomics data. And we did the EWAS, and initially we just found tons of hits that actually overlapped with the MQTLs, like FATS came up, NUT8, and all that stuff. It was a bit frustrating, and we said, what is going on here? And probably what I think, I mean, it could be just a coincidence. There could be a SNP being neighboring a CPG site, and it could just be LD. There could also be, of course, functional things. There could be a change in the metabolite. There could be back regulation, and then methylation regulating that. But in any case, once you have a SNP behind that, it's not the environment that's driving this. But then we said, let's correct out all of these SNPs and say what stays. And then if you go for the things that stay, and that's something initially also I was like a bit like, okay, it makes sense. So you find a lot of CPG sites that co-associate with vinyl sulfate, and that's a metabolite of smoking, and all these are smoking CPGs. So that was kind of a positive control. And there was another one, we really never really understood what it was. I recently had a look at it, could be alcohol consumption, but not really sure. But the one that's really interesting is this block here. And again, there's lessons learned. At the time, I just listed the top hits. And I think it's a bad idea just to list top hits with metabolomics because you miss what's all beyond. So I thought this was unrelated hits. we just reported them. But then later, and I think especially the study of John Chambers with BMI and with diabetes and obesity, they actually highlighted also some of these, especially TXNP come up very often again with diabetes. So we took the occasion of a small study we did here in Qatar on Arabs with type 2 diabetes to revisit the data. And what you see here is actually the summary data from the supplement of the Patterson study we did. And here are the three loci, APCG1, CPT1A, and TXNP. They are all three diabetes and obesity associated sites. But now if you look at the metabotype here, there it comes. Here, it's diabetes-specific metabotypes. You find all these markers, the Biocrates platform we found with diabetes, the proteomics, the lipoproteomics data, and also this very well-known alpha hydroxybutyrate from the metabolome platform. And the thing is, they all three had the same metabotype. So the message here really is like, look at the metabotypes, not only on the top hit. So that's something what statisticians sometimes omit. All right, and then naturally, of course, we can do the same thing with proteins. That's something I did more recently with Shazza Sakhlul from my group. So we again went to Cora, and in our QM-DIAB study, we had replication. And I think the message here is the problem why people don't like methylation so much is there's so much of a confounding. For genes, you know, it's always the genetic variant that is causal, but not for EWAs. It could be, or in many cases, is the environment. So what we did here is we regressed out step-by-step potential confounders like sex, white blood cell counts, SNPs, age, smoking, BMI, diabetes. And I just want to show this. It's not an animation, it's just successive slides, but this would be the Manhattan plot if I just do proteins against CpG. And you see enormous p-values, high degree of inflation, a lot of stuff above the genome-wide significance level. The moment I throw in sex, you see there are already lots of hits that disappear. And then if I put in white blood count, I mean, then really things disappear a lot. And that's standard protocol to actually put in, sorry, I say white blood count, I mean the Hausmann coefficient, so the different blood parameters here. So there's a lot of confounding between CpGs and different blood types. And that really see, if I flip this back and forth, disappears then. And then if I take out the SNPs, actually, you see the change in scale. So what really disappears when you take out the SNPs are these very strong hits that are actually driven by genetics. The rest remains, and then I can take out age. So you see there's still a lot of age confounding here. And then the other things, smoking, BMI, and diabetes, actually they don't change much. They are more local things. There may be tens of loci or something like that. So with that study, we then said we can combine this with all kind of other data. So with the proteomics and the methylation association, we do clinical phenotype from Cora. We have metabolites from our QM-DIAB study. We have gene expression from public databases, and then go to IPA for more information. And this really what works nicely is when you now integrate this. So this would be one of our associations. It would be like association between 3 CpG sites, especially NLRC5 here, with 7 different proteins, and they are all replicated between Cora and QM-DIAB. And now I can enrich this, and I actually find a lot of associations in Cora with LDL, HDL, metabolic syndrome, body mass, even type 2 diabetes. Then I add metabolites, and I find, in this case, I find them from urine, in neptarine, especially neoptarine, which is an inflammation marker. And then I can go to bias that shows me there's also transcripts associated with that, and connect the genes and the proteins using IPA, using the connect function. And then I think it's already clear for people who know the proteins, but if you don't, you see, for instance, here HLA, MHC class 1, actually B2M is a constructive part of the MHC complex. So in the end, what this really means, it's a complex network that links NLRC5 methylation, which is the methylome, low-grade chronic inflammation. And if you go for this literature links, there are now EWAS catalogs as well. So you find many of these CpGs are actually associated with diseases that have a very strong effect of chronic low-grade inflammation, especially rheumatoid arthritis, lupus, and you also have a CVD disease here. Okay, so again, for this, we have a table. So that table isn't that full yet, but I think it will take off. And with this, I think I should also come to my conclusion. So coming back a bit to what I'm presently doing. So this is work I do here in Qatar. We have a study here we started in 2012. And we try to throw on that all omics platforms we can. There are about 350 samples, and we had the Biocrates platform that were actually measured in Munich, the Metabolon platform, Genomics, Nightingale, Lipidizer, and then we have genotype, we have methylation. Now we have RNA-seq as well, Exicon circulating microRNAs, the proteomics data I showed to you. We have proteomics, protein glycation, N glycans and IgG glycans. Now we moved also on, added the OakLink platform, and most recently, we also brought metabolomics platforms to Qatar, initially one from Metabolon, and now we're also setting up the new 500 kit here at the Hamad, together with Anya Halama. All right, and then really very quickly, just to show you, so this is really a prototype. I just couldn't stop myself showing it. It's really something I just did the last few weeks. The thing is now the data from here, I can link this in all ways. We can do GWAS, we can do EWAS, we can do transcriptome-wide association studies, we can do GGMs. You've seen that earlier as again inside of these platforms. And then what Anya is looking is also what we call the mutual best hits, like the orthogonality between the platforms, which metabolite actually matches which metabolite or which protein. And for that, so I really just want to show you a screenshot on that. But for this one, you can now go and say, I want to query, for instance, the Biocrates kit, hexose, and then it would find hexose here, and then it would find around all the other hits around. So here's the hexose, and then you would see, like here's the glucose, it's a mutual best hit on the Nightingale platform. If you go deeper down here, I think it's a bit hidden. There's the TXNP hit that goes with diabetes, HbA1c, H association with proteins. In this case, there's no SNPs. But I think this is the kind of thing, we have this as a prototype for 350 individuals, but imagine you would have this kind of data coming from the NACO or from UK Biobank or the Japanese studies and all these things we've seen today. I think that would be really, really huge. All right, I think I ranted a lot just to do a little bit of self-promotion here, just to give you a reference to where you can read things up. So I was lucky to work a long time with Christian Giger. So in 2012, we actually wrote a review on metabolomics, a lot about study design. And more recently, we reviewed also genetics against proteomics. But I think much of what is said here is also true for metabolomics studies. With that, I want to thank my funders, Qatar Foundation, my research team in Qatar. And of course, there's so many people, I cannot list them all I've seen in the past. And here, my last slide, the yellow arrow here shows Zurich. So my team, when I left Munich, they sent me this photo, and I think you see a lot of people here. I think Anya there's Zurich here. I think Gabi is somewhere here. I think you will spot yourself. All right, with that, thank you very much. I think I'm done here.
Thank you, Karsten.
So I hope that came through. Sorry for the buggy Zoom. It wasn't the network. It's the Zoom. I don't know. I had this just yesterday one.
So I think we have understood you very well. So thank you very much.